Hello and welcome to your fourth video here that will be on adaptive immunity as a part of our Chapter 22 video lecture series. So just to review, our adaptive immunity is going to involve our specific lymphocytes that are going to respond to an antigen. So they are going to first make contact with an antigen and cause lymphocyte proliferation. When I say proliferation, I mean that there is a rapid multiplication of these cells to increase in number. And so that will help with our immune response, and they'll secrete certain products that will, again, amplify that immune response. And as we mentioned before in previous sections, they will have a longer response time. It takes time to tailor-make that immune response to a specific antigen. And since it takes days to develop, they are considered our third line of defense. Remember that the first two lines of defenses were within our innate immune response. And so within our adaptive immune system, we have our T cell mediated or cell mediated immunity. And in this will involve our T cells. We have different types. We'll get into the specific actions that they take. And then we have our B cell immunity, which is also known as our antibody mediated immunity, that will involve our B lymphocytes, our plasma cells, and the antibodies that they secrete. So first, you heard me mention the term antigen many times in that that previous slide, so we need to define it. Essentially, an antigen is going to be a marker on any cell. So our pathogens are going to have these antigens on them. Here's an example of one. You could see that we're representing the antigen as different shapes here, and they can have thousands of these different types of antigens. And it is made up of a protein or a large polysaccharide. And I want you to dig back to some of the terms we learned about in microanatomy, such as our protein capsid, that is the outside shell of our viruses, or the cell wall of our bacteria or fungi that we talked about in the first section of this chapter. Some of our bacteria have specific toxins that they are going to secrete, and other times we in our body will have abnormal proteins or tumors that will have antigens on them that our body will want to trigger an immune response to as well. So we can classify our antigens as being our foreign antigens or non-self antigens and our self-antigens. And this is pretty self-explanatory. Our foreign antigens differ from our own body's molecules, and so we're going to find them on pathogens, or maybe we have some type of transplant and we'll see the cells there as foreign. Our self-antigens will be found on our own body cells, and so we should not have any of our immune system recognize this and trigger an immune response. If that does happen, then that is how autoimmune disorders begin. We start having a reaction to our self-antigens and seeing them as foreign. Now, to further complicate things, we have antigens on these pathogens, but on the antigen itself, we are going to have these antigenic determinants. These are also known as epitopes. So I really liked this figure here that shows an antigen and how that on that antigen we have multiple different epitopes or antigenic determinants. This really helps it be a little bit more complex and specific, so that we are going to be able to, again, tailor-make antibodies that will attach to those specific epitopes. So we're only combating that foreign molecule that we have. Now let's do a clinical view on the general causes of our autoimmune disorders. Our immune system lacking tolerance for our self-antigen essentially produces these autoimmune disorders. We're creating an immune response to that, and it could be due to cross-reactivity, which is what's pictured over here, essentially meaning that when the immune system recognizes two different antigens as being similar and responds to one as if it were the other. We can see here in the image that this antibody here has that hemicircle shape to it or semicircle shape to it, and it's meant for this structure here with this antigen. But over here we may have a reaction to this because it has the circular structure as well. We may also have some altered self-antigens that can be perceived as being foreign, and we can enter areas of immune privilege. And what we mean by that is that immune privilege is a mechanism that is going to help protect certain tissues in our body from an immune response, which allows them to tolerate antigens without triggering inflammation. For example, we have rheumatic heart disease, type 1 diabetes, multiple sclerosis. I'll just speak to the type 1 diabetes in that we have what's called islet protection. If you remember, our pancreas has these islets of Langerhans that are the ones that are going to produce things like insulin and glucagon. And so our islets can be thought of as mini organs within our pancreas, and they are producing insulin when we have an increase in glucose within our blood, and they're protected from immune cells by a connective tissue network and some extracellular proteins. And this barrier gives these islets immune privilege. Next, let's get into the general structure of our lymphocytes. And if you look at either a T cell or a B cell, they're all going to have these unique receptor complexes, and about 100,000 per cell. So that's quite a bit. As we mentioned before, they're going to have specific antigens on there. And also, if you have a T cell, you will have a T cell receptor, which you can see down here on our helper T cell and our cytotoxic T cell. And then on our B cells, we of course have our B cell receptors. And we're going to utilize some of these receptors to make contact with the antigen. So our B cells are the ones that make a direct contact with the antigen. With our T cells, they have to be presented the antigen by another cell. So within these cells that present it, the antigen is going to be processed and presented. The T cells then have co-receptors. And these are our CD proteins, as far as the ones that we're going to talk about, that will help to facilitate the interaction. So sometimes you may hear our helper T cells being called CD4 cells, and that's because they have this CD4 protein on them near a T cell receptor, and they are going to work to help in the cell-mediated immune process, and they help our B cells in the antibody-mediated process and within our innate immunity. For instance, they are able to activate our NK cells and macrophages that work within our innate immunity to go through phagocytosis, or for our NK cells to be able to release granzymes and perforins. With our cytotoxic T cells, you may have heard them be called CD8 cells, and that's because they have their CD8 protein that works with their T cell receptor in order to become stimulated. And once they're, I should say, activated, they release chemicals that will destroy the other cells, so a pathogen. We do have some other T cell types of T cells, like memory T cells and regulatory T cells. We'll talk about them a little bit later. So let's focus now on that antigen presentation. These are going to be cells that display that antigen on their plasma membrane so that our T cells are able to recognize it. We have two types or categories of cells that can present an antigen. So that is going to include all of our nucleated cells that are able to do it. So essentially everything except for, let's say, our red blood cells, because we know they don't have a nucleus. And then we have our antigen presenting cells. And I have this on another slide, but our antigen presenting cells would consist, oh, actually I have it on this slide, would consist of our dendritic cells, our macrophages, and our B cells. So again, they are able to present the antigen, and they help to activate our helper T cells and our cytotoxic T cells. And so to help present this antigen, we need to produce a MHC molecule, or a major histocompatibility complex molecule, and place that antigen on it. So what these are transmembrane proteins, and we'll see that visually on the next slide. We do have two different types that you need to know for this class. MHC1 is found on all of our nucleated cells. So I always remember this as the saying, all for one and one for all. Okay, they just kind of go together. Whereas our MHC2 molecules are going to only be found on our APCs, our antigen presenting cells, in addition to them also having MHC1 molecules, because our APCs are also nucleated cells, so they're going to have both. So first, let's talk about how our nucleated cells are going to synthesize and display their MHC class 1 molecules. These MHC class 1 molecules are glycoproteins, and they're genetically determined and unique to individuals. So it's almost like a fingerprint. And that's why when we do things like transplants, we have to make sure that we have a close enough match. And so we are going to continually synthesize and modify within our rough ER here, and then have it move through our Golgi apparatus and eventually move through secretory vesicles to be then displayed on the plasma membrane here. And these at this point would have a self-antigen on them displayed, since they are a part of our body. But if we were to, let's say, become infected by a virus, then we're going to want to take one of the antigens from that virus and display it on the cell to signify that I've been infected by a virus, and you need to trigger the immune system in order to destroy me so that the virus cannot go and infect other cells. Now let's take a look at how MHC class 2 molecules are displayed on our antigen presenting cells. So these will also be glycoproteins. They are going to be synthesized and modified by the rough ER, and then they will go through our endomembrane system. So they'll go through the Golgi apparatus and move into our transport vesicles. Now here's where we get a little bit of the phagocyte process taking place. We know that here is a exogenous antigen, meaning that it has come from outside the body, whereas when we talked about, let me go back here, when we talk about endogenous proteins, it means it's a part of our body. It was produced inside our cell. So we pull in that bacterium here, and it is now called a phagosome. This phagosome is going to fuse with our lysosome, and we now call it a phagolysosome. And so now our secretory vesicle that contains those MHC class 2 molecules will have that foreign antigen displayed at the end of them, and we are going to want to move this vesicle through exocytosis to be on that plasma membrane so we can display that foreign antigen. And in this way, we can communicate with our helper T cells and basically activate them so they can alert our immune system. So here's a nice visual figure of an interaction between the receptors of our T lymphocytes and their MHC class molecules. Here's our T cell. You could see that we can have either CD4 or CD8 markers there. And here's our T cell receptor that is coming into contact with the antigen on the MHC class 2 molecule here. This is a nice little close-up here when we have a CD4 protein, and here's an example of when we have a CD8 protein. As a clinical view, I kind of mentioned how when we have transplants that we need to match for the MHC molecules. We are going to first see that transplant as being foreign, right? It could be a kidney, liver, or heart. And that's why we test before to make sure that we are going to be as close as possible to the MHC molecules. And of course, we also want to match for blood type. Otherwise, our innate and adaptive immune systems will try to destroy that transplanted tissue, and the recipient's immune system will be suppressed with drugs so that we can try to prevent that from happening. Okay, and the last thing we are going to cover is an overview of life events of a lymphocyte. So we've got 3 main events. First is our formation of our lymphocytes. They're going to first be produced or originate within our bone marrow, which is a primary lymphoid structure, and then will travel depending on what type we have, right? So if we have a B lymphocyte, it stays in the red bone marrow so it can mature, and the T lymphocytes will travel over to the thymus in order to mature, and they'll be able to recognize any specific foreign antigens. The second event is called activation of lymphocytes. And so at this point, the B and T cells will travel to secondary lymphoid structures. So we covered this in the last chapter. It could be our lymph nodes, our spleen, the tonsils, and here they are going to be exposed to an antigen and become activated. And once activated, we need them to proliferate and clone themselves so that we can form identical lymphocytes and essentially create an army to combat a specific antigen. The third event is called the effector response. It's the actual action of our lymphocytes to help get rid of this antigen, which belongs to a pathogen within our body. So our T cells are going to migrate to the site of infection, whereas our B cells stay in the secondary lymphoid structure and start to create plasma cells, which also are going to proliferate, that are in charge of synthesizing and releasing large quantities of antibodies. And they then, the antibodies, that is, will travel over to the infection site through the blood and the lymph so they can combat the pathogen.